Okay, so in this section, we're going to go over, this is mainly chapter 20, and we're going to talk about, um, basically, we're going to talk about hormonal control, um, which is here, and we're going to kind of work our way through the nephron and talk about what's absorbed where and and how that kind of relates back to how urine is produced and the concentration of urine, whether we're going to have very dilute urine or whether we're going to have very, very thick concentrated urine. Okay, so we're going to go over each one of these. Diuresis, which is a term, and that's where the term diuretics come from, which is a drug that makes you produce more urine, or a drug or a hormone, as we'll see. And then we'll talk about a hormone called aldosterone, and then a hormone system called the renin-angiotensin pathway, which also uses aldosterone. And then we'll talk about another one called atrial natriuretic peptide. Now, there's another one that I don't have listed here, which is called vasopressin, which is also called antidiuretic hormone, or ADH, that we'll talk about. Okay, so if we look at the tubule, so this is your nephron, and remember we have the glomerulus surrounded by the Bowman's capsule, which makes it the thing called the renal corpuscle. And so as blood is filtered here, it's basically, at first, it's just kind of blood plasma because it's the same concentration, everything is the same in terms of ions. The only thing left out of what actually makes it into the nephron, so the filtrate, the only thing left out of that are large blood cells, well, any blood cells, and platelets, and large proteins. Okay, so those are the only things that don't get filtered through here, and so it's essentially just the liquid part of blood when it first starts out. now, now we don't want, because we have like a fourth of our blood is going to the kidneys, we don't want to just excrete that as urine, because you see this is a one-way tube, and the only true exit that it has is urine. So we don't want to urinate out all of our water and electrolytes and everything, because we would die in a couple of minutes. So we have most of it is reabsorbed. About 99% is actually reabsorbed. 80% of that actually happens in the proximal tubule. So what does that mean? That means most, or 80%, about 80%, you read different things, but it's a very high number, is reabsorbed in the proximal tubule. Okay? So that's where most of this reabsorption takes place, is right here. Okay? And that includes ions, that includes water. We talked about that in the previous section. It starts out with active transport of sodium, and then chloride follows. Calcium goes, water, because all the ions are leaving and out here, then the water leaves, and little by little, all of this stuff just kind of goes out. And remember, it moves into the paratubular capillaries and then back into the blood. Now, things change a little bit when we get into the loop of Henle. So in the descending loop of Henle, we have this area here where water can move out, but salts really don't. Okay, there's a little bit of movement of salt, but primarily what's moving out of here is water. Okay, so that's reabsorption. When I say moving out, I'm usually talking from the perspective of the nephron, so that means moving out means reabsorption. So water is moving out, and then we get to the other side, the ascending loop, and guess what leaves there? Salts. So sodium, chloride, potassium. These things, oops, chloride is negatively charged. Sodium, chloride, and potassium move out, but no water. Okay, so water can't move out. And that's kind of a special property of nephrons. Usually we think of water being able to go wherever it wants to, and that's true in most of the cells, but in the renal tubules, in the nephron, that's not the way it is. They have tight junctions that don't permit water. That way they can control, because otherwise if water just moved out, we would not be able to control our urine concentration. And we can see that we can control that concentration anywhere from 50 milliosmolars being just a measure of how concentrated something else, to the high number, which is 1200, which is very concentrated. So we can control that, and we do that partially by controlling how much water leaves, which we control that over here in the collecting duct primarily. Okay, so that's a place that water can or it may not leave. Okay, And so that really gets us to the next point. So the descending loop of Henle, water moves out. The ascending loop of Henle, salts move out. And then the distal tube and the collecting duct, distal tubule, you don't really know. So salt, mainly sodium, may or may not move out. We just don't know if it's going to move out or not. And that's really determined by how much salt is in your body. Do you need to conserve salt? If you need to conserve salt, then yes, you will reabsorb it back into the body. If not, then you won't. It'll just continue on. And the same thing with water. Do we need to conserve water? Or if you need more water in your body, then you will insert these things called aquaporins, nice name, which means water pore. You'll insert these little water pores, and the water will be able to move out. Okay, but it's determined by hormones. So how we say that is the distal tubule and the collecting ducts are fine-tuned by hormones to increase or decrease water and or solute reabsorption. But again, going back to the first parts, the proximal tubule, the descending limb, and the ascending limb, those are pretty much set. You have water moving out over here. You have salts moving out over here. Okay, so let's just look at this for just a second. Because one thing that we know is the cortex, the renal cortex, is very dilute. Okay, and the medulla is very concentrated. Okay, so if that's the case, then we know that somehow these salts have to be moved down. But if we look at this and if we don't know that it's very concentrated in the bottom part and very dilute in the top, we would look at this and we might say, oh, look at all the salts over here, with all of these salts over here, because they're being kicked out, actively kicked out, and then the water over here, which is being allowed to leave, then we might say, oh, well, it looks like it's going to be very dilute on the left and very concentrated on the right. But that isn't the way it is. Do you kind of understand that? Because this is a very important point. The salts are over here and the water is over here, and yet when we look at a kidney, we see that it's very salty down here and very dilute up here. So that means, and it's correct to assume this, that somehow all of these salts, as they're being removed, are being pulled back down. Okay? And that means this water, as it's being removed, are being pulled back up. Okay? And that has a name. And it's called the countercurrent exchange, or the countercurrent mechanism. Okay? And it's called countercurrent because, look, this is our descending loop. This is the water that's leaving, okay? And it's going from around 300 milliosmoles, and it sees this higher concentration down here, and we put these little aquaporins in there so it's able to move. And it as it's coming down, it says, oh, well, look how concentrated this is, and this is a little thing called the vasorecta, okay? Now, the vasorecta is just a capillary that's moving in the opposite direction. So this is kind of a loop. I don't really know where to begin. If I just say, oh, look, it's 300 milliosmoles here, but it's more concentrated here, so the water moves out to try to offset that, right? It moves through osmosis. It's being pulled to where the more concentrated area. But then as it moves in, it's immediately swept upwards by the vasorecta. So it moves into the vasorecta, and the vasorecta is going in the opposite direction. It's going counter in direction to the direction of the tubule. This is going this direction. This is going up. So when this water moves out and moves into the vasorecta, it's immediately pulled back up. Okay, so let's look at this side. So we have, this is our renal tubule. These are the salts, the sodium, potassium, and chloride, that are being kicked out. And as they get kicked out, they're salts. So it's 600 milliosmolar here. So it's more concentrated here. So they look up here and they say, oh, it's only 500 there. I kind of want to go there. But as soon as they get in there, they're swept down. Okay? And so if we look at both of these things again, the water's moving out to the higher concentration, to where it's more salty, and then it's immediately swept up. The salts are moving out, trying to go down their concentration gradient, and they're immediately swept down. That keeps the water moving up and the salts moving down. And that is why you have a high concentration down at the bottom and a lower concentration up at the top. And in fact, these salts that are being kicked out, actually, they're actively being kicked out, will actually cause a concentration that's even lower, around 100 milliosmol or even lower. Okay, so let's just concentrate for a second on the movement of these salts out. So if we kind of move away, we can say, okay, yeah, water's leaving here. We can accept that. We know that water moves down its concentrate, or it's down, it moves up its it moves into a more concentrated area through osmosis. So it's going to move to where it's more salty. So it makes sense that all you have to do is put a pore in there and water will move. However, these salts have to be actually forced out. Okay, so the salts have to be moved out of there, and they do that through this little pump. It's called the sodium potassium chloride transporter, or the NKCC, sodium potassium chloride transporter. And so it moves those out, and then those are immediately taken up by the vasorecta. I know I'm drawing it on the outside. It was drawn on the inside before. It actually loops around, and then those are brought back down. Okay. Now the reason this is important is because you're probably going to see this again if you've ever heard of a drug called Lasix. Okay, not the eye surgery, but Lasix. It's a blood pressure drug, and it's in a category of drugs called loop diuretics. Okay, and it, by moving the salt out of the tubule, so moving the salts back into the body, basically, is what you're doing. Okay, and it'll go back into the blood, back into the extracellular fluid. What you're doing is you're actually causing water to also want to move out into this area. You're conserving salt in the body, and if you conserve salt in the body, then you're going to have water around that salt. And so it actually raises blood pressure by pumping these salts back into the body. More salt in the body, more water in the body. Okay, so what these drugs do, like Lasix, is they block this sodium potassium chloride cotransporter. By blocking that, they end up actually keeping the salts in the urine, which means that the water is less likely to leave. So the water stays in, and you have more urine production, which means you have more fluid in the body. Okay, so that's why we talk about this. All right, so let's talk about overall in general water balance. So the kidneys conserve volume but can't replace volume. Now so the kidneys are able to say, oh, okay, let's get rid of this amount of water today, and then the next day maybe they say, well, we're not going to get rid of as much water. So they can conserve volume, but they can't replace volume, and we kind of know that anyway because that's why we drink. Okay, so there's this term diuresis. So let's say we have a whole lot of water. So let's say we're drinking very, very well, and we have a lot of water in a day. I'm not sure what kind of bottle that is, but let's say that's water. And so we're drinking a lot of water, and so our body is going to say, okay, we've got way too much water, we need to get rid of it. Or it might say, oh, the blood pressure's a little high, so let's get rid of it. That term is called diuresis. Now, diuretics are drugs that we take. We've probably heard the term diuretic, that promote the excretion of urine. So that means things like caffeine. Caffeine might cause us to urinate more. Alcohol causes us to urinate more. And that drug that I was talking about, Lasix. Lasix is also a diuretic. Okay? It causes a higher volume of urine, and that's because it's leaving the salts in the urine, which means that water is going to stay in the urine. Okay. So again, diuresis, the removal of excess water in the urine. All diuretics lead to an increase in urine production. Yes, we've mentioned both of these things. And now let's talk about how they have their effect. Okay, how do diuretics work? We already talked about Lasix, that it blocks that sodium potassium chloride pump on the ascending loop of Henle. But they also have other ways to either control sodium in the distal tubule, so sodium reabsorption in the distal tubule, or maybe how much water reabsorption in the collecting duct. Okay, so that's another way that they do it. And we're going to talk about 3 or 4 different ways that these diuretics could have their effect. Okay, and like I also mentioned, certain diuretics are used to treat high blood pressure. And usually they aren't very expensive. Okay, they're usually cheaper and they are nearly as effective. They've done studies where they took some more very expensive high blood pressure medications and compared them to the efficacy of just using diuretics, and they found that diuretics were actually just as effective. Okay, so let's talk about a hormone called vasopressin. We've mentioned that before. It's made by the posterior pituitary. If you remember talking about the posterior and the anterior pituitary, this is supposed to be a pituitary. There's your pituitary gland. Comes down from the hypothalamus, remember that? And the posterior pituitary makes vasopressin. Okay, this vasopressin, what it does is it causes the collecting duct. Okay, so the collecting duct of the nephron. So there's the descending, ascending, distal, and then the collecting duct. So this area right here of the nephron causes it to be permeable to water. Let's stop and think about what that means. Remember, it's very, very concentrated down here, high concentration down here, 1200 milliosmoles. By inserting, and this is what it does, is inserts something called aquaporins. Okay, inserts those into the wall of the collecting duct and allows water to leave. Well, of course, water is going to leave as it gets more concentrated down here. So it sees this concentration of 1,000 and it's only 800 here. So yeah, it's going to leave and it's going to go to that higher concentrated area because that's what water does. That's osmosis. Okay, so all you have to do is give it a channel to move through. Okay, and that's what vasopressin does. Now, I will say that alcohol blocks vasopressin. Okay, so it blocks the effects of vasopressin, so it doesn't allow that water to move out, keeps it in, and then you have very dilute urine. Okay, so if you aren't able to remove that water, so if you block vasopressin, then you have very dilute urine. Okay, so this is kind of a picture. The vasopressin causes the insertion of water pores in the collecting duct. This is kind of what's happening. There are these little storage vesicles. That's where the aquaporins are stored on the inside. And then when vasopressin comes down and binds to its receptor, it will cause a signal to be passed, a second messenger signal to be passed, and it'll insert these water pores. And as soon as that happens, all of a sudden water can just leave. Okay? It can just leave. So there are a lot of drugs that will block this. Okay, they'll block that process, which means that the water can't leave, which means the water goes out as urine. Okay, so more urine production, more water left, or more water leaves the body, lower blood pressure. Okay, so that's what this question is asking. If vasopressin, which as I mentioned is released by the posterior pituitary, increases the number of aquaporins, what effect would this have on blood pressure? Your job here is to remember, if this is the tubule, okay, your job here is to remember that the aquaporins are letting the water out, so it's causing reabsorption. Okay, so that means that if the water is going back to the body. Okay, so if vasopressin increases the number of aquaporins, then what's that going to do to blood pressure? Well, what's the effect of more aquaporins on reabsorption? It increases reabsorption. More back to the body, more in the body means higher blood pressure. That means that vasopressin increases blood pressure. Okay? How do body fluids, so more water back in the body, how do body fluid levels affect blood pressure? Increases it. Okay, so now there's another hormone called aldosterone, and what it does is it puts sodium channels, let me see if I can get a better color that's a little more easy to see, but it puts sodium channels in the distal tubule and sometimes in the collecting ducts, And that allows, actually pumps sodium back to the body. Okay, so when aldosterone, which is a hormone, is released, sodium goes back to the body. So that's reabsorption. Okay, so more sodium is reabsorbed. So aldosterone is released by the adrenal gland, endocrine pathway responsible for the regulation of sodium. Um, takes place in the distal tubule and first part of the collecting duct, which we see here. Higher aldosterone levels lead to increased reabsorption of sodium. That means it stays in your body, so it leaves the tubule and goes back to your body. Because the sodium potassium pumps are pumping potassium in. Oh, potassium and sodium in opposite directions. It also has an effect on potassium. Okay, so it's actually the sodium potassium pump that's doing this. So that means the potassium is being pumped in. But just concentrating right now on the fact that sodium is leaving, what does that maybe mean for blood pressure? That means that because sodium is moving out, you're going to want to retain water. Because remember, where there's sodium, there has to be water around it to keep that osmolarity proper and correct. Okay, so we want to have the right osmolarity. So that means if we're retaining sodium, we're also retaining water, which means that more fluid, which means an increase in blood pressure. So that means aldosterone, the release of aldosterone, causes an increase in blood pressure. Okay. So just to kind of give you an idea of how this works, I mentioned before that when aldosterone is released, it's working in the distal tubule, so descending loop, ascending loop, distal tubule, and collecting duct. It's working in this area here, okay, the end of the distal tubule, first part of the collecting duct. And if this is the inside of the nephron, well then what's happening is sodium is being moved this direction. And here's our sodium potassium pump. Potassium is being moved in this direction. Okay, so this is kind of a problem. A lot of diuretics that will stop this. Okay, so there are diuretics that block the effects of aldosterone, and a lot of times what they do is they cause the potassium levels to build up because usually this thing is working and sodium is being reabsorbed. But if we decide we want to take a drug that blocks that reabsorption, then what ends up happening is that we also will start maintaining, keeping more potassium in, which isn't necessarily good. Okay, but this whole slide is just to sort of show you how the aldosterone works. It's a hormone, so it's moving through the blood. It binds to its receptor, and then it starts putting in new channels, new sodium channels, new sodium potassium pumps, and it gets that sodium to be reabsorbed. Okay, so that's what you need to remember. It increases reabsorption, okay, in the distal tubules and the first part of the collecting duct. Okay, so if you have, so let's ask this question. This is a fair question. What effect would the release of aldosterone have on blood pressure? I just answered that actually. So if we have sodium in the distal tubule being reabsorbed, that's what aldosterone causes to happen, then we need to have around that water. If we have more water, this is associated with the body. If we have that, then we have an increase in blood pressure. So I go over that because that's an important thing, and I'll ask that a couple of times on the exam. Okay, so we have this big fancy system that is actually started in many ways by the kidneys themselves. We call it the Renin-angiotensin aldosterone system. Okay, so these are hormones, okay, here. Now, renin is actually, if I draw a nephron again, remember, if this is renal corpuscle, here, let me draw this again. Okay, I'm going to draw this a little more accurately the way that it actually shows up. So if this is your Bowman's capsule and glomerulus, what ends up happening, this is the proximal tubule, and then you have the descending limb. It actually goes this way. Instead, we usually draw it to go that direction, but it actually goes this way, and it goes right past the afferent and efferent arterial, and then you have the collecting duct here. So it's constantly measuring itself. So you have your, I'll draw it in a bit of a different color, so it's not very accurate, but here's your afferent arterial and here's your efferent arterial, okay, and it goes in there and it makes the glomerulus. This is constantly monitoring what's going on in there. And when your blood pressure gets too low, it might release a molecule called renin, okay? And this renin, so let's just kind of go through this list. Renin is released when the juxtaglomerular apparatus senses low blood pressure, okay? So this is the kidney itself kind of saying, oh, well, there's pretty low blood pressure. So you have this going around in your blood right now. You have this molecule called angiotensinogen, okay? It's floating around in your blood right now. It's made in the liver, and it's not doing anything. It's not active. It doesn't really do anything until renin is released, and then this angiotensinogen will turn into something called angiotensin 1. Okay, you can already tell what's going to happen here and what the purpose of it is because it's called angiotensin. Angio stands for heart, tensin means pressure. So it's obviously going to affect blood pressure. And it was released because blood pressure was low. So angiotensinogen in the blood will turn into, after renin is released, will turn into angiotensin 1. Angiotensin 1 doesn't really do anything either. So angiotensin 1, if it has the number 1 behind it, it kind of implies that there's going to be an angiotensin 2. Okay, so we have another molecule floating around in our blood plasma all the time called angiotensin converting enzyme. Angiotensin converting enzyme is what causes angiotensin 1 to turn into angiotensin 2. Okay, and guess what? That's pretty much it, because angiotensin 2 has a whole lot of effects. It does a whole lot of things. One of those things that it does is that it leads to the release of aldosterone. This is probably very confusing right now. But if you think renin is released by the kidney, and then what does that do? That causes, ultimately, that causes aldosterone to be released. And we just talked about what aldosterone does. It increases reabsorption of what? Sodium. Okay, so that means more sodium in the body, more, we don't need that S there, more sodium in the body, which means increased blood pressure. Okay, so that means the release of renin makes sense when low blood pressure is sensed. It converts inactive angiotensinogen into angiotensin 1. Angiotensin converting enzyme then turns angiotensin 1 into angiotensin 2, and angiotensin 2 leads to the release of aldosterone from the adrenal gland. It's very complicated. It involves a lot of different parts of the body. You've got the liver involved. You even have the lungs involved. Okay, so some other things that angiotensin 2 does, besides, it also causes the release of vasopressin. It increases thirst. Yes, aldosterone from the adrenal cortex. It causes higher increased cardiovascular response, so it causes the heart to beat faster. It causes vasoconstriction. It's one of the strongest vasoconstrictors. So it does everything. This angiotensin 2 does everything it can to increase blood pressure. Okay? And yes, there are drugs that block this. Remember this thing called angiotensin converting enzyme that converts the angiotensin 1 into angiotensin 2? We have inhibitors for them. They are called, oops, not sure where N came from, but they are called ACE inhibitors, because angiotensin converting enzyme is shortened to ACE. Okay, So angiotensin converting enzyme, or ACE, converts that angiotensin 1 into angiotensin 2. If you block ACE, you block angiotensin 2, which means you block aldosterone and you block this other thing, vasopressin, that we're getting ready to talk about. Okay, so let's look again. Angiotensin 2 has many effects. It leads to the release of aldosterone. That's the one we're mainly talking about here. It stimulates thirst. It's a vasoconstrictor. Increases sympathetic output of the heart, so it increases stroke volume and heart rate. Okay, so it really works to increase blood pressure. So that's why things like ACE inhibitors work so well, is that they don't allow that angiotensin 1 to turn into angiotensin 2. Okay, so what if you inhibit ACE? If you inhibit the ACE inhibitors, then you're going to have lower blood pressure, or at least not an increase in blood pressure. Okay, and you're actually going to slow down that system overall. Okay, so another one, there's this term natriuresis. That's how you say that. Natriuresis is the loss of sodium and water in the urine. Okay, so that means that instead of being reabsorbed, okay, so this proximal tubule, descending loop, ascending loop, distal tubule, and there is the collecting duct. So that means that instead of sodium being reabsorbed, this will cause sodium to be excreted. Okay? If you excrete the sodium, let's think about that. If you excrete the sodium, what's that going to do to your blood pressure? Well, remember, where the sodium is, the water has to be, the water has to be around it. Okay? So if we excrete more, sodium, we're going to have more water being released too. Higher urine volume, decreased blood pressure. This is the only one, remember, because aldosterone increased blood pressure. Okay, so aldosterone, angiotensin 2, those both increased blood pressure. Vasopressin increased blood pressure. Okay, remember it was putting more water pores into the cell. So vasopressin increased blood pressure. And so this is really the only one that we've talked about so far that is actually decreasing blood pressure. Now, it's interesting, and it's easy to remember why if you remember how it's activated. Okay, it's called atrial. Oops. For some reason I want to turn all my A's into N's. Atrial natriuretic peptide. Sometimes it's called hormone. Okay, or hormone. When your atrium indicates heart. So when your heart stretches, okay, so it's on the atria of the heart. When that stretches a little bit too much, so atrial stretch, then that will release this atrial natriuretic peptide. The atrial natriuretic peptide we care about, it does all of these things. It also has an effect on the hypothalamus, the adrenal cortex, medulla, but we mainly care about this one. And what it does is it increases GFR. Okay, we haven't talked a whole lot about that, but if you increase GFR, that means you're pushing fluid through more quickly, fluid's entering the nephron more quickly, and then you produce urine more quickly. So you increase urine, which means that you decrease water in the body. Okay, so that's going to lower the blood pressure. So your atria in the heart stretch too much. It's too much fluid, too much blood, stretches too much. So what do you end up doing? The quickest thing you can, and that is simply get rid of that extra fluid. Okay, and this is what this says. Okay, so natriuretic peptides, it also blocks renin, it also blocks the aldosterone, but it's produced in the atria of the heart upon stretching, and then they enhance sodium, and therefore if you enhance sodium excretion, the water has to stay there too, and so it also enhances water excretion. Released in response to increased pressure in the atria, increases GFR. So that's how it has its method. It just sends things through more quickly, so that sodium doesn't have as much of a chance to be reabsorbed. It stays in the tubule, and then it's released as urine. Okay, so it also suppresses or inhibits these other things. Okay, um, so what have we gone through? We've gone through. Let's go back and revisit aldosterone for just a minute, because remember we had the sodium potassium pump. And remember, if we're going to cause reabsorption, I know this is confusing, but if we are going to, if this is the distal tubule here, this is the collecting duct, if we are going to cause sodium to be reabsorbed, we're going to lose potassium. Okay, so aldosterone, we've been talking about it in terms of what it does to sodium, but it also kind of has the side effect of potassium, and this can be a problem with some diuretics, as I said. So we call this hypokalemia, okay, when we have low potassium levels, and hyperkalemia when we have high potassium levels. So really what I'm most concerned is that you understand that the sodium potassium pump pumps things in different directions. So if we decide we want to reabsorb sodium, well, as a default, we're sort of getting rid of potassium. Or if we decide we want to get rid of sodium, one of the things we're going to do is we may have elevated potassium. Okay, so don't forget that when you're in practice, or if you ever take a diuretic, or if you ever give a diuretic, you know, that's why you're checking the labs. You're checking these potassium levels to make sure that even though you're controlling the sodium in the body, that you're not doing something that you don't want to happen with the potassium. So you'll have to keep an eye on that. Okay, behavioral mechanisms. Drinking replaces fluid loss. It's this, in other words, one of the things that happens here is we become thirsty. That's a fantastic way. You're sure we can just dump everything, but or we can conserve as much water as possible, but unless we put more in, it doesn't really help us much. So drinking replaces fluid loss after a big meal. A lot of times after a big meal, we get very, very thirsty. I know I do. And so oops, which is not what this says, although that's true. This is saying that if you have low sodium, and this is fantastic, if you don't have enough salt in your body, it will actually stimulate a salt appetite. You will actually crave salt. Okay, that's not something I notice a lot of times until somebody puts, you know, some fries or potato chips in front of me, and I just eat them because I had no idea that I wanted salt so badly. But also a lot of times it just makes us hungry. So it may not even be the salt appetite. It might just be appetite in general. And then we also have avoidance behaviors. If you know, we live in Kansas, or I live in Kansas, if you drive down the road and you see, you know, on a hot day, a lot of times you'll see cows standing in the pond or under a tree, or, you know, we're always seeking shade, that kind of thing. Desert animals avoid heat. Okay, so those kinds of things. Those are behavioral mechanisms. All right, now the last thing we need to talk about is acid-base balance. We've talked a lot about this. We've seen this equation here in some way or another. H2O, CO2, carbon dioxide plus H2O makes H2CO3, which then can be broken down into this right here. Let's just sort of read through this, because what's really most important is that, let's look at this picture. It says acidosis. The pH of the blood, and therefore the extracellular fluid, has fallen. It's become very acidic, very low pH. So one of the things that's going to happen by the kidneys is that it will simply dump the hydrogen. Okay? This is going to go into the urine. Goodbye, hydrogen. You're going into the urine. Part of this equation, this reaction, is that hydrogen is made over here, but bicarbonate is made on this side. And it says, oh, bicarbonate, we're going to pump you back over here. And as soon as bicarbonate gets pumped back over here, it will find a hydrogen, and it will turn into, whoops, I thought there was a picture of it. It will turn it into the H2CO3. Okay? And that's kind of our blood buffer. So what's really going on in the kidneys is that When your pH is low, it will simply dump hydrogen. Okay, so we can reverse this if the pH is high. We can reverse this and it will actually dump bicarbonate and it will move hydrogen in this way. It's a nice little system. It's very direct, very straightforward what happens. Okay, so normal pH of plasma is 7.38, 7.42. You might see 7.35 to 7.45. It's very closely regulated. Changes have huge effects on proteins. Um, Abnormal pH affects the nervous system, acidosis, neurons become less excitable, CNS depression, so that everything just sort of slows down. The opposite with alkalosis. Alkalosis means a high pH. It becomes alkaline. Okay, and so if it becomes alkaline, we see that neurons are more excitable. Okay, so these things definitely have an effect, and it's a very, very tight range. So there are a number of things that can cause this. The release of fatty acids, amino acids. That's why a high protein diet tends to cause your pH to run a little bit low. Okay, lactic acid. So that comes with like exercise. If you exercise very intensely, a lot of anaerobic exercise, then that's going to produce lactic acid, and that lactic acid is going to make your pH a little bit lower. Okay, but fortunately, mostly what we have is we have our blood buffer, which takes care of things. We also have proteins that will absorb those extra hydrogens to a point. If it's too much, then those proteins will stop being functional. And then we also have ventilation, so we can breathe faster. Remember, as we increase our CO2, we also, so as CO2 levels rise, pH goes down. Okay, so increased CO2 means a decrease in pH, a decrease in CO2, which would happen if we breathe faster. We're breathing off all of that extra CO2. Then that's going to increase the pH. Okay, all right. So if we look at this, let's look at it in a more organized way. pH homeostasis, to maintain that pH in that tight range, the first step are buffers. Okay, so that hydrogen is either going to combine with something or be released, and that's going to control the buffer. So it's going to control that hydrogen ion content. And the main buffer, yes, of course, is the blood buffer, which is this carbonic acid. Okay, and it can go in either direction. And so it can either go in this direction where you get rid of the hydrogen, or it might go in this direction where you actually produce more of the hydrogen. Okay, so there's a lot. So it works in a certain range. But then, you also have, like I said, proteins. Proteins are very instrumental in soaking up that extra hydrogen. Their phosphate ions and their hemoglobin. We didn't talk about those. And then ventilation. That's a very rapid response. Okay, so when you're not able to maintain, when this buffer isn't working and your hydrogen levels are getting to be a little bit high, well, then we might breathe a little faster, which throws off the carbon dioxide, and your hydrogen will go away. It'll move that in the other direction. And then, of course, the one we're talking about now, which is renal. It's the slowest of the three. It directly excretes or reabsorbs hydrogen, and it can also affect the bicarbonate ion. Okay, so indirectly by changing the rate at which the bicarbonate buffer is reabsorbed or excreted as well. Okay, so one thing I want to just mention very, very quickly, and that's the Micturition response. Okay, so this has somatic sensory. Okay, so this means something we are aware of. And it's also parasympathetic, which is our fight or flight response. Okay, so what am I talking about? I'm talking about when you have to pee. And you have stretch receptors that send a signal that says, hey, our bladder is getting large. Okay, so we've been pumping urine into it. Our bladder is getting large. It's starting to stretch. We have stretch receptors that then go back and will tell, this is what it's going to do. Okay, we know we can override this, but that stretch receptor is going to say, hey bladder, time to contract. and let the urine out. But at the same time, that's what this is saying, at the same time it's sending a signal up to the brain and saying, yes, no, should we or should we not? And the brain might send back a message that says no, and then you won't urinate. Okay? So this is a reflex, and once your brain says, oops, once your brain says yes, then that releases any kind of inhibition it had, and this process will take place. It'll say stretched, okay, we're going to contract, and we're also going to release this sphincter. Okay, there's an internal sphincter and an external sphincter, and we can consciously control that external sphincter, and we can say, okay, let it go. Okay, so what that means is, and that is a parasympathetic response. That's a very automated response. So the parasympathetic nervous system is the rest and digest. So it sort of makes sense that that's when it would be activated. Is that gets activated when we're very calm. Okay, and so that process gets activated, and we try to override it with the brain. Sometimes when we get scared or something like that, that overriding doesn't take place, and everything just sort of stops, okay, and we lose control over this external sphincter. Sometimes this external sphincter may get weakened, and once this builds up to a certain size, it may push through already, and that's incontinence and things like that. But overall, it's a very simple reflex. Stretching causes contraction, overridden by the brain, we hope. Okay, so that's it for the section. I actually think the Micturition was probably in the previous chapter, but I wanted to talk about it very, very briefly. Okay, so that's it for this entire section. Thank you.